From Microbe TV, this is Office Hours for Wednesday, the 8th of January, 2025. I'm Vincent Racaniello, and welcome to your viral corner of the internet. Welcome, everybody. Let me extend a special welcome to our moderators tonight. We have Vanity Nutrition, Tom Steinberg, Les Fabi, Steph SF. Thank you for being here and moderating. Tell us where you're from, folks. What do we have planned for you tonight? Two main things. First, put up a new Beyond the Noise today. And YouTube took it down. They said I was spreading medical misinformation. I'll get to that. I am furious, beyond furious. And also we have a mini lecture on chronic wasting disease, which is developing some interesting things these days. So we'll check that out. But before that, let's find out where everyone is from, because you are Office Hours. Without you, it's nothing. Without you, it is nothing. Wayne is in Urbana, Illinois. 15F minus 9C. What is it here? Minus 6C feels like minus 15. Pretty cold. Darn cold. Patty is in Massachusetts, where it's 18F and windy. Will is in Changsha, China, 3 degrees C, where it's morning. Yeah, fires are really bad in Southern California. I was just watching them. Oof, terrible, huge, really bad. Hello, Mark Martin from Puget Sound, Washington. Tacoma, Washington, University of Puget Sound. Good to see you here, sir. It's 21C in your office. Yep. Martha's in Charleston, South Carolina, where it's 41F. Hello, Rima from Iowa. I know where you are. Hello, welcome. Angela's from upstate Manhattan, 15 blocks north of the GWB. GWB is about 178th, if I'm not mistaken. In Iowa, it's close to minus 15. Oh, my gosh. Kang is in Chicago, where it's minus 3C and cloudy. Chris writes, Chris is from Montreal or thereabouts. Happy New Year. Good luck with your new president in his drive to rule North America and promote anti-vaccine propaganda and white supremacy philosophy. Yeah, that sums it up very nicely. You're absolutely right. Hello, John from Eden Prairie, Minnesota. Yeah, we'll keep you company. No worries. Mark says it's hard to see the fires in SoCal. I was born and raised there. I've checked in with all the people I still know, all okay, but so tragic, indeed. Indeed. Abdul Aziz is from Kingdom of Saudi Arabia, where it's 5 degrees C. Maureen is in a cold northeast Ohio. Hat, LA, 66 and smoky. MN is outside of DC. Nebby is from Central Florida. Jonathan is in Encinitas, where it's 19C and dry. Soul Symphony is from Egypt. Wow. Whoa, I don't recall anyone from Egypt here ever. Welcome, Soul Symphony. Hope you enjoy. Brian is in Montreal, minus 11 and snowy. Pete Glass is in London. Vincent can do all the Racanielling he likes. Oh boy. Yeah, this is a real problem. I'm very frustrated, but I'll tell you about it in a moment. Hello, Lise from Columbus, Ohio. Al is in West Virginia. Mood Indigo, great name, is in India. We're at 17C. Rima writes, I'm concerned bad people, anti-science will come at you. Yeah, it's a possibility, but I'm going to keep doing what I'm doing. I was talking to Paul Offer yesterday. He said a lot of universities are just afraid to say anything. They say, let's just keep quiet. I think that's the chicken thing to do. It's not how things get done, right? Huh. Julie S. is from central Pennsylvania. Rad writes, I was watching Beyond the Noise and it just stopped. I thought you took it down yourself. No, they took it down. The schmucks who don't know anything at YouTube. Well, maybe it's an algorithm. But if you here's a clue. If you go to Microbe TV, you can still play it there because it's embedded. And it still works. They haven't actually taken it down. They've just made it invisible on the YouTube pages. Andy is in Mornington, Australia. Welcome. Brian is in Bridgeport, Connecticut. Claire is in the UK. John is in Tallahassee, where it's 42F. Wow. Very warm. Well, not so warm. Warmer than here. Cheryl is in Santa Rosa, California. And Rima says in Iowa it's minus 10. Rad, Toronto, minus 15. John watched the Beyond the Noise before it got taken down, but furious they would take it down. Yeah, I'll tell you about it. Just hang on. Let me get through. Hi from a very cold West Virginia. One thing, folks, I do not deal in misinformation. I may make mistakes, but I do not do medical misinformation. So they don't know what they're doing. Hello, Jefferson Space from Miami. Good to see you. And Carol, our favorite nephrologist from Bakersfield, California, where it's 56 degrees Fahrenheit. Hans is in Ecuador. Thank you for joining us. Pat is in Nova Scotia. Logotic is in Chicago, where it's minus C. Doreen is in Hartford. Abdur is in Buffalo tonight, minus 9C. Actually, I'm home tonight. It's, for a few weeks I did office hours at the incubator, but it's late. It ends at 10, right? And then I have to make my way home. It's just, in this cold weather, I'm not doing it. So I do the office hours here. Wendy is in Pleasantville, New York, where it's 18 degrees C. Tracy is in Chapel Hill, 27F. Maureen must be in Ohio, right? James is in Longmont, Colorado, 28F. Ontario, Canada, Cheryl, not the 51st state, minus 14C. Here to listen to rants about our collective future. It's just frustrating as hell, right? Margot is in Phoenix, 57 degrees. Ariozone is in bitter cold and blustery Asheville. Feels like 5F. Abdur is also from Egypt. Hey, you two guys can chat. That's cool. Or two people. I don't know if you're both guys. Hello, Patricia. Massachusetts. Kip from San Francisco. Thank you for your support of science education. Vincent, 61F in SF. Laura reports the ED is full of flu sufferers. Yeah, not surprised. Dean is in Wisconsin. This gentleman is safe in Santa Barbara, but it doesn't have a name. Soul Symphony is from Egypt, but moved to Minnesota for a virology PhD. Very cool. Prion is a former medic from Romania. Welcome. You're here every week. I recognize that. Maharsh Kim. Bird flu. Yeah, they do that, don't they? Mike and Lori are from balmy Seattle, 44F. And Mark writes, Vincent listens to all my venting. I will listen to all of his, and I know I am not alone in this. Hey, nobody has to listen to my venting, but I usually save my vents for a good cause, right? And there are multiple causes. You want to get me to vent? Just say SARS-CoV-2 did not come from nature. I'll vent, because that's the most ridiculous statement you could make. Not the most. There are many ridiculous statements. March Kim is in Calgary, where it's 38 F3C. And yes, Eman, you got it. What I convey is all science-based. Yeah, for sure. Yeah, the thing is a little off to the side. I don't know why. We'll move it over. Things, technology, you know. Let's move it back. Okay. Bad Cat is in Winthrop, Maine. 15 degrees feels like 0. Tom Hardy is from Salt Lake City, 35F. So people are out jogging in shorts and T-shirts. Pete is in London. It's just a degree above freezing in the heart of London. Definitely below freezing in the outskirts and colder in the Midlands. Marie, good evening to you as well. Welcome. Marcelo is from Brazil. Welcome, Marcelo. Sabrina is in New Jersey. Welcome back. I know you found us last time. It's a good place to be here. John is in Port Townsend, Washington, where it's 46F. Pete missed last week's to wish happy birthday to me. Final night in Malta, had to pack. Yeah, the second was my birthday. So I'm now a year older, 72. I'll just check the Beyond the Noise podcast list. It says it was updated yesterday, but the most recent video is unavailable. Yeah, they took it down. I'll tell you in a moment what's going on. SRR is in Paso Robles, 56F. NY Cohen, Westchester, New York, feels like 4 degrees Fahrenheit. You're welcome, Breestar. I'm happy. That's what I do. I'm a teacher. I was a scientist for many years. Still am. You know, to be a scientist, you have to be a teacher to some extent. But I'm the world's teacher of virology, Earth's virology professor. Okay. So let's put up Microbe TV. I'll put the page here and I'll share it with you. Let's get it here. Okie dokie. So I will share the screen here. Okay, so this is the Microbe TV webpage, right? And you, let's go to our, do we have Beyond the Noise here? No, it's not here. So we'll go more shows, and here's Beyond the Noise. So number 48 is just published this morning. You can actually, oh, now it's gone here. The embed is gone as well. Bastards. Real bastards. Actually, it's working. You just have to click it. The thumbnail is not there, but what they said is they took down your video because it's spreading medical misinformation, starting at 4 minutes and 25 seconds. So if you play this, what Paul says at 4 minutes and 25 seconds is, vaccines work. There's no question about it. Okay? So they're saying that that is vaccine misinformation. Now, I've already appealed it. I don't know what's going to happen. But what burns me up is that RFK Jr. has been on multiple podcasts on YouTube, including Lex Friedman and Joe Rogan, saying medical misinformation, and they don't even ding them a minute. Not a minute. They never take any of that crap down. Okay? Can you imagine taking down an episode of Rogan or Friedman? Because on Friedman, RFK said vaccines have not been shown to have any benefit. They're not safe nor of benefit. He said the polio vaccine kills more people than it protects. So, in fact, this video, which had gone up today, is questions for RFK Jr. Senate confirmation. It's based on Paul Offit's blog post. And I went through the questions with Paul. And the public needs to know this. None of it is medical misinformation, YouTube. So I don't know what's going to happen to my appeal, but I don't know how to go beyond the appeal because this video has to be seen. Okay. And I'm supremely frustrated, as I said, because so much misinformation gets published on YouTube and nothing gets done about it. And here I am trying to provide real information and I get dinged for it. Do you know how frustrating that is, folks? I can't begin to tell you. Anyway, here, let's look at his blog post. You said no vaccine is safe and effective, okay? You said the polio vaccine killed more people than it helped. You said the COVID vaccine was the deadliest vaccine ever made. You said you encourage people not to vaccinate their babies. These are all documentable things, okay? And that's part one. And then part two, you said no vaccine is safe. This is still part one. Yeah, I got part one here by mistake. All right, so let's go to the index. Damn. No thanks. I'm already subscribed. Part two. Oh, he's got two part ones. Here we go. Sorry. You question whether vaccines cause autism. This is misinformation. You state that vaccinating children with COVID vaccines is unethical. You state that both hydroxychloroquine and ivermectin work to treat COVID. Wrong. You say the hepatitis B vaccine does not work. Wrong. This is all medical misinformation. Spewed by RFK Jr., nominated to be the head of HHS. What kind of a screwed up country is this that a guy like that, who says nothing of truth, gets nominated for that position? It is, of course, it's more frustrating than my video getting taken down, but we don't have to go there because we all know how screwed up that is. So, you write to YouTube and tell them to put the video up because it's not misinformation. Maybe if they get enough emails from people, I don't know who to write to. But, I think someone complained. Yes, Marco says, Marco reported the video for misinformation. So what if I report Lex Friedman's video for misinformation? I'm sure many people did and nothing happened. It's a waste of my time to deal with this, but it has to be done because people need to hear this video because the confirmation hearings are coming up and he needs to be asked these questions. Anyway, if you want to see the video, go to microbe.tv/BTN. You can see it there. So Rad says, I think someone just reported it. I posted a TikTok of flu shot RCTs reducing heart attacks. I appealed and they fixed it. So I appealed. Hopefully they'll see the light. I suspect that they don't even bother to investigate when someone complains. But how does this work that Rogan and Friedman don't get dinged? They don't get pulled down ever. Why? Why not? How do you report? Because I'm going to go and report those two podcasts with RFK Jr. as spreading medical misinformation, okay? You should all do that yourselves. Come on, folks. We need to work. Please. Okay. Let's go back to the to the comments. So what is Vanity? According to YouTube, you have 30 days to submit appeal. So I did. There's no submitting an appeal. All you do is click a button that says appeal. You can't write anything. You just appeal, and you have to depend on them looking at it. So senior reviewer who did not make the original decision will review the request. It will be either upheld or reversed. This is unfair, isn't it? That video needs to be up. There's no misinformation in the video. What should I do? Yes, Elizabeth, you're right. Friedman and Rogan make too much money for YouTube. They'll never get taken down. This is just really unfair. John writes, this is obviously overreach by Google, an example of why you should consider including discussions of AI on Microbe TV. Oh, we started to do some papers where AI is prominent, yeah. Yeah, it's very funny that what was I going to say here? Yeah, often is a vaccinologist, an MD and a vaccinologist. He says vaccines work, and that's dinged as misinformation. Why would anyone at Google even listen to someone who counters the statement that vaccines work? You'd have to be an idiot to think they don't. Oh, and I know a lot of people say they don't work, like RFK Jr. and others. Yeah, if they say that, they're idiots. I'm sorry. End of story. And it's terrible to be ad hominem. I agree. But you're not speaking from sincerity when you say vaccines don't work. No, RFK Jr. didn't hear that video, but one of the people who love him, someone wrote in the comments, doesn't matter what you think. He's head of HHS already. He's not head of HHS. It has to be confirmed, and he might not be. Best thing that could happen is he might not be concerned. Yeah. Anyway, it was a good video. You can still watch it. Go to microbe.tv. Let me put the link here in the let me put the link in the show notes, in the chat, so you can go listen to it. Watch it if you haven't already. Assholes. Jesus. My nice thumbnail. Okay. Here we go. Here's the link. Check it out. But really write. Find someone to write to and include the link to the video and say, this is not misinformation. This is truth. Put it back up. I do need your help for this, okay? Because we're small. Put the transcript up. Steph says. Well, yeah, but YouTube gets a lot of views, right? A couple of new people have shown up. Let's see. I did Paso Robles. Bryce stars in Fredericksburg, Virginia, snowy and 20F. 28F. Marco Mont, 22F on the east end of Long Island. Side Teach 84. Hello from Ventura. 60F. Longtime listener and lurker, first time chatter. Welcome. Bernard, welcome. Waving from DC tonight. Greetings from North Carolina. Thank you very much. Yes, you could read Paul Offit's Substack. But you know, a video is another way to reinforce the point. So, I mean, I could put the audio up, but it's not going to help. We'll see what happens. Put up the transcript on Blue Sky. You mean just post a link to it on Blue Sky, right? Yeah, I know. Cheryl, the video is not live. Yeah, it's live on the website. Just click it. Well, I just did. Let's see if it's broken on the website now. Because maybe if they get too many links from the website. They'll turn that off too. Let's see. Yeah, for me, the website link is still working. Just click the play button in the middle. The thumbnail's not showing because they took the thumbnail down, but it still plays. So Vinity says you can write an appeal. Well, when I went to the entry for this removal, it just said, did you want to appeal this decision? I clicked yes, and thank you. We will get back to you. But I'll try the YouTube Studio start appeal. I will do that, yeah. Thank you for that, Vanity. And everyone go click the Rogan and Friedman interviews with RFK Jr. Please do this for me. Or not for me, for science. Go on YouTube, find the Rogan and the Lex Friedman podcast with RFK Jr. and then click on it and say it's misinformation. Please, we can get 100 or 200 people doing that. I think it would make a difference. Yeah, video removed for violating YouTube terms, which they say it's spreading medical misinformation. Martha writes, China and Russia pay people to spread misinformation. Maybe they just discovered you. Well, this is a hotbed issue, right? The whole RFK Jr. Confirmation. And many people want him confirmed. So there's going to be a big effort to silence people who speak against him, which is really disgusting, right? I didn't grow up that way. You tell the truth. You don't get smarmy behind the back. JM is in Germany. Thanks for all your work. Helped me tremendously during the pandemic. Good luck with all the political issues that are ahead in the U.S. I suspect that what happens is that if they get a complaint, they just take it down. They don't even look into it because they don't have the time. I don't know how that could be automated, or maybe it's automated. And if you hit a complaint, it's taken down. But then Rogan Friedman, what's the story? Maybe they have special dispensation from the Pope. Hello, Jessica, also from, I want to say Ohio, right? Toledo. Welcome. I will write, I will make a transcript and put it on Blue Sky. Yeah. So Tom says, in the past year, I've had multiple posts on theoretical math get removed by Facebook. Why? Why would theoretical math be reviewed? Okay, Rays, we need help with you to do two things. First, go to YouTube and Microbe TV and say the video Beyond the Noise 48 was taken down. You should be able to get a link and tell them there's no medical misinformation in that video. It's all truth. And then go to the Lex Friedman and Joe Rogan podcast with RFK Jr. and complain that that's medical misinformation, because it is. And mine is not. Mine is information. Hello, Charles. When you say RFK is not sincere, you're overestimating his intelligence. Yes, you're right. People lie with purposes, yeah. Yeah, disinformation can spread like a virus. This is disinformation about our video, yeah. Heather, I'm so sorry you and Pull Offit's video got labeled. I will write them. Yes, please, everyone write YouTube and say this is not misinformation. I've heard it. Sabrina slashing away the disinformation online like a machete through the Jeep jungle that it grows at a ferocious rate. Yes, after absolutely. Anyway, Ozone writes, as of yesterday, Mark of Meta is removing all fact checking on Instagram and Facebook. Seems ironic that YouTube would fact check you and PO for correct information. It's horrible. Yeah. Oh boy. Paul Offit. Yeah, Paul Offit is a primary source of medical information. Abdur says, with the level of false propaganda coming out, do you think we will get through this before significant losses until the public consensus rebuilds? Well, I could be really negative and say we're going to have a huge loss in confidence in science and public health and vaccination, and we'll see a resurgence of infectious diseases. And, it may take a year or two, and then maybe calm heads will say, wait a minute, what are we doing here? It's just crazy. I can't think about it. I can't think about it. Yeah. Okay, let's see what else we have here. There was a question here from Luna. Luna's reading the paper of Dr. Sinusi, how she identified HIV. I did not understand the experiment. I don't have it at my hands here, right? Let's see if we can find it quickly. Okay. So according to ASM, 1982, doctors gave Montagnier's lab a sample taken from a patient with generalized lymphadenopathy, swollen lymph nodes, which is early stages of AIDS. She took that sample and put it in cells in culture. And 2 days after, she detected reverse transcriptase in the supernatant of the cells in culture. So HIV, a retrovirus, retroviruses are unique in that they have an enzyme in the virus particle called reverse transcriptase that converts the viral RNA to DNA so that it can be integrated into the host cell. So she devised an assay for reverse transcriptase, and she used that to measure the cells 2 days after infecting them with the lymph node sample from the patient. And she said, there is a retrovirus in this culture. And so then you could take it and take that culture medium and use it to infect new cells and see if it propagates. You could get more samples and see if you get the same thing. You can do electron microscopy and go, this is a lentivirus, a certain kind of retrovirus. And eventually you can do the genome sequence and show that it is a retrovirus, a new retrovirus, HIV-1. And you can then develop assays to check patients to confirm that they all have the same virus. So, but originally she infected cells in culture with an explant of the lymph node and showed that they were infected with a retrovirus, right? Okay. Dean wants to know, how will quantum computing affect the field of virology? I don't know. I mean, is quantum computing on the cusp of being here? I just don't know the field at all. What's the advantage of quantum versus the computing we have now? If you tell me that, I can give you some ideas of how it would affect the field of virology, but I just don't know enough about it. I don't know what it's called when short segments are posted. Perhaps make short segments. Yeah, I can do that. It was like It was 30 minutes long. We could make 15 segments of 2 minutes each. That's a good idea, Marco. Thank you. I appreciate that. Hello, Costello from Ohio. Don't worry. We're just getting started. We are just getting started. All right, so I have to make shorts, and then I have to go to the website, YouTube Studio, begin review, review reason, start appeal. Okay. Vanity, would you do me a big favor? Just text that to me so I don't forget it. All right, Hungry Ghost says, I got 3 vaccines a couple of days ago. Can't get RSV until I'm 75. No, I got RSV 2 years ago when I was 70. So that's not true. I don't know what country you're in, but you should be able to get it. It's the CDC recommendation. Marion is in Ireland. We have lots of snow in the southwest. Schools are closed. -4 in Virginia, Cavan. I have tuned to your podcast for tuned to your podcast for years. Thank you, Marion. I hope you enjoy them. 162 people right now. Please go and report the RFK one right now, especially the part where he says no vaccine is safe and effective. Yes, please click the like button, folks. We have 170 people, 95 likes. Click the like button. We need to get near 170 likes. That would be good. Yeah, University of Toledo in Ohio. Great. I will remember it eventually. So one of your critics of the math post said it incites violence. This is just BS. And the reviewers don't even bother to check. Heather says, I get so angry when I hear anti-vaxxers act like they're being suppressed. And they don't want you to know when their videos get insane high numbers. Yep. They're not being suppressed. It's freedom of speech. They can publish their videos, but they shouldn't be allowed because they're spreading misinformation. Anchi, Bakersfield, Trumpland, California. Bakersfield is in California, right? Spring with 61F. Vanity, can you upload the video to Blue Sky? I don't know. Is there a length thing? 30 minutes. I don't know. I can check. Do you know when the Rogan video dropped? I don't know. Just RFK Jr. Rogan. He's the only one. He was only on once. Armoda, when is the 6th edition of your textbook coming out? Not for a while. Not for 2 more years. 2027. We are working on it at the moment. If you need it, just get it now because it's going to be a while before it comes out. Okay, John. So, yeah, how is the SV40 promoter different from the virus? So the promoter is a small DNA sequence in the viral genome. The SV40 is a DNA virus, and a small part of the DNA has a promoter, which you can take out its own and put into another gene, and it will drive expression, the synthesis of mRNA of that gene. It was never used in polio vaccine. SV40 was a contaminant of polio vaccines in the 50s and 60s. In the 60s, the oral poliovirus vaccine was contaminated for a while until they realized this. And so people were getting infectious SV40. And so people were concerned that this would lead to cancers. And so many epidemiological studies were done, and there was no increase in any kind of cancer in people who received the oral polio vaccine. So it didn't cause any cancers. That's what RFK Jr. was referring to. He said tens of millions of people got cancer from the polio vaccine. It's not true. And of course, very shortly thereafter, SV40 was removed. So since the 60s, there hasn't not been SV40 in any polio vaccine. And it works really well to prevent poliomyelitis. Yep. Raise 11. Will do. Thanks for posting your class. Didn't buy the book. A little expensive. Not sure I would pass a final exam, though. But I always think that way about my chances. You can find it used somewhere for sure. I bet. Yeah. Okay, Rima, I'll look at the paper. But I think what I told you is basically what they did. Okay, but I will look at it. And Vanity says, if you'd like the book, try here. Abe Books. Yeah. Martha writes, I will contact YouTube also. Today, the coward Mark Zuckerberg decided he will no longer fact-check posts on Facebook or Instagram, as we already noted. It's just disgusting that one person can have such control over social media and dictate crap like this. So now you can say whatever you want. So I should post the video on Facebook. I don't know if I could post a 30-minute video. Can I? Can I post a 30-minute video on Facebook? Or I can split it up into two 15-minute videos. And I'll try Blue Sky as well. Yeah, but YouTube is the big one. Tom says quantum computing has been at Los Alamos for years. They actually have a networked set of quantum computers. Not sure what they use their network for, though. Okay. Big YouTube makers get a free pass. Smaller ones don't. Yeah, we're small. We're very small. But we are in the top 5% of YouTube creators, right? With 134,000 subscribers. But we're not huge. I would like to be huge so I can just get around this crap. I work really hard. We work really hard to give you the truth. And this bugs me. It bugs me. It bugs me. Marge, glad to see so many folks here tonight. Thank you for continuing to share your time and knowledge, Dr. R and Team TWIV. You're welcome. I love doing the podcast. I love doing these office hours as well. It's a great chance not just to teach you things and answer your questions, but to hear what you're thinking about. All right? And tonight, of course, it's timely. I got to complain about something and ask for your help. So it's great. I am in Tennessee. They must have changed the rules. Okay, let's look it up. RSV vaccine recommendations. CDC. Yeah, the states can do their own thing. You're absolutely right. Oh, that's very interesting. Look, I'm at the CDC site. RSV vaccines are recommended for all adults 75 or older and adults 60 to 74 who are at increased risk for severe RSV. So you can, as I say, your state may have its own rules because the CDC are just guidelines. Remember that line in Pirates of the Caribbean? They're not really rules, puppet. They're just kind of regulate. What would he call them? Because she wanted to do a parley. And they say, not really rules. They're just kind of suggestions. So the CDC makes suggestions, and the states usually follow them because they don't have any way of making their own suggestions. But in this case, 75 and up, unless you have some kind of medical condition, then 60 to 74, you can go down as low as 60 years old and get an RSV vaccine. But you can go to CVS, a drugstore. That's what I did. I just walked in and I want an RSV vaccine. Okay, they didn't ask me anything. They have my age there on their paperwork. And they said, sure, sit down. Boom. That was it. So, and of course, I didn't have to pay for that. My insurance paid for it. So if you're worried about that. So that's what I can offer you. Angry Penguin. Can you explain the origin of the term toll-like in toll-like receptors? You're the best science presenter at YouTube. Oh, thank you. Thank you. But now I'm taken down, so I must not be so good. Toll-like receptor came from, in the 80s, two scientists in Germany were studying fruit flies, and they were making mutations to try and identify genes involved in development of the fly. And one day, Eric Wieschaus said to Christian Nusslein-Volhard, he said, "Come over here and look at this weird fly." And she looked at it in the microscope and she said, "Toll," which means far out or wow, wild, something like that in German, right? Toll. And they called it the toll gene, and it stuck. Now they're all those class of genes are toll-like receptors. Yeah, that's where the name comes from. Nothing to do with paying a toll. Yeah, yeah. Tom, thank you so much for doing that. I really appreciate it. Yeah, and you're right, Hungry Ghost. So you're 60, 70, 74. If you have some issues, or you just go to a drugstore, they're not going to ask you anything. Tryon wants to know if coronaviruses induce cold agglutination syndrome or co-infections with mycoplasma. I don't know. I've never heard it. Don't know, Tryon. Frank is in Santa Barbara. Hello, Frank. John says, I think the Rogan video was over a year ago, and that is on Spotify. It's not on YouTube. There are some accounts that repost clips or portions, I think. All right. Well, certainly Lex Friedman's interview with him is there, and you can ask for that to be taken down. Do you recommend getting a Novavax vaccine for a healthy young male? Well, I'm not going to give you medical advice, Touge 410, because people get mad at me. And they're right. So let's look it up for the CDC recommendations. CDC Novavax recommendations. That usually helps. Clinical guidance for COVID-19 vaccination. So people 60, so you're a young person, so this doesn't account for you. These are for old people, and these are interim. So complicated. All right, so the Novavax is authorized for people 12 years and older. That's cool. They don't particularly prefer one over the other. So vaccination is recommended for everyone over 6 months of age. Okay. And they recommend people get all COVID vaccine doses. So when they come out with a new variant-specific vaccine, they recommend that you get that, even if you're young. Yes. So CDC says you should do that. Okay. So I didn't give you medical advice. CDC did. Yep. Oh, has there been any official comment from CDC about the misinformation spread by Kennedy? No. The federal agencies are all silent. All silent. Not surprising, right? So, Hungry Ghost says the pharmacist denied me the RSV. I'm sorry, Hungry Ghost. She told him you have some lung condition. Make something up. How's the pharmacist going to know? Tell me you have asthma. They're not going to know. That's what I would have done if I really wanted. But here they just gave it to me. Yeah, influenza is on the rise. As we saw from Kip in San Francisco, the ER that his wife works in, flu is on the rise. But yeah, this is the season for flu now. Yeah. Is it true that washing your hands is better at sanitizing it for norovirus? Yes. Purell alcohol-based sanitizers do not work. They do not inactivate norovirus. You have to wash your hands with soap and water for at least 30 seconds. And what that does is the soap will remove the debris, including viruses, from your skin, most of it, and then the water will wash it away. It's not actually inactivating the virus, it's removing it from your skin, which is just as good. And it's probably not 100%, but it doesn't have to be 100%. Will, thank you very much. I appreciate that you have done that. And I hope everyone else can send YouTube feedback that Beyond the Noise 48 is based on verified science. Maybe we can get a crowdsourced revolt and get it reposted. Yeah. Thank you very much. What's the fuss over the rise of human metapneumovirus infections? Anytime there's a rise in infections with any virus, there's a fuss, right? You know that, Pete. It can be any virus, norovirus, human metapneumovirus. It's driven by the press, including YouTube TV news or whatever that is, and TV, that whatever people get their news from, social media. Big outbreak of norovirus. So what? What are you going to do? There's nothing to do about it. If you're vomiting and have diarrhea, you stay home until it goes away in a couple of days. And next time, wash your hands before you touch your mouth. It's very straightforward. When I go out and about in the winter, I always wear gloves. Always. It's a really good way to prevent you from contaminating yourself. And you may say the gloves get contaminated. Yeah, but I'm not going to put a glove in my mouth. I might bite my finger, but I'm not going to put a glove in my mouth. Why did it take until 2001 to discover it? There are lots of viruses that cause respiratory infections. And for the most part, people don't bother to diagnose which one you have. We know the more famous ones like rhinoviruses and coronaviruses, adenoviruses, paramyxoviruses. And so only when one rises above the noise does it get recognized. And that's what happened in 2001. Here you go. Where can we view all the videos and audios you've been collecting? The ones, a number of them are on YouTube Shorts. Some of them are on TikTok. So YouTube has most of them, but there's still a few missing. Okay, that we have to get to, but we've put up a bunch of them over there on YouTube. Go under the Shorts column, right? You can go to our page and look under Shorts because that's where they're classified. I'll show you. Why not? We got YouTube being a jerk. So, and let's see. We're going to turn this on screen share. So here, you see down here below my picture, you got home, you got videos, and shorts. If you click shorts, you get all the shorts that we have posted. And there are a lot because we take excerpts from all our podcasts. But here's Tom's little testimonial, right? It's Tom's Paul Kramer. Also my opinion on the nominees for these various positions. Here's Betty's. Here's Kip and Laura's. Here's John's. So, that's where you can find them. All there. Lots of shorts. People seem to like these shorts. Okay. Back to the questions. John writes, is there a difference between PCR and RT-PCR? When I see NAT, is that a general term for PCR? So a NAT includes PCR and other methods of nucleic acid amplification. That's what NAT stands for, nucleic acid amplification test. Because PCR was the first, but there are others now, like LAMP and others. So NAT includes all of them. PCR is when you take DNA and you amplify it by polymerase chain reaction. RT-PCR is when you want to know how much RNA is around. So you take reverse transcriptase, the RT, and you turn the RNA into DNA, and then you amplify the DNA by PCR. So in the old days, when you took a nasal swab and you wanted to do a PCR for SARS-CoV-2 RNA, you would take the sample, extract the RNA, convert it to DNA, and then do the PCR. That's why it's called RT-PCR. Twitch. Post a video on Twitch. Yeah, this is good. This is good advice. So we've got all the social media sites. Basically, I'm going to try posting them. Blue Sky's video limit is 60 seconds. Facebook's is 240 minutes. Facebook, here we come. First thing in the morning, I'm putting it on Facebook. Thank you. SRR Joe Rogan Experience number 1999, Robert Kennedy Jr. There you go. Go to it. Tell them it's medical misinformation. Hello, Jill. Wilmington, Delaware, where it's 23 and cold. Margot says influenza has spiked in Phoenix. Not surprising. Not surprising. Is human metapneumovirus a concern? Yes, it's a concern for people who are at risk for severe infections, right? If you're young, let it rip through you. You're going to get nice immunity. There's nothing you can do. There's no vaccine. There's no antiviral. Just stay home and don't spread it. But yeah, if you're older, it's a concern. It could lead to pneumonia. So we don't want you to do that. So what do? Well, if you're elderly in the middle of the respiratory season, you should consider using a mask. I do. I ride the trains. I use a mask. I don't want to get sick. If someone says to me, sir, are you sick? I said, no, I'm trying to prevent getting sick. Get out of my face. I wouldn't say that, no. But I would say, no, I'm trying to prevent myself from getting sick. But no one has ever commented on my mask. It's my glasses. They're scared of them. Thank you, John, for your contribution and support of science communication. You see, we need help. We need your help. Not only financial, but we need your help to combat this misinformation. Thank you very much. I can't find who mentioned Los Alamos. That was, I can tell you who it was. It was Tom Hardy of Maths. He mentioned quantum computing and Salt Lake City. So let's get back to Margot here. I hear that, and my first thought is I too glow in the dark. I just tell people it was the tritium in the water. I have childhood friends still at Los Alamos. Vincent, do you have a lecture or talk on the channel about the history or origin story of viruses in general? Yeah. Virology lectures number 1. And in a couple of weeks, yeah, there you go. So Pete's telling you exactly what I said. In a couple of weeks, there's going to be a brand new one. It's not going to be much different, but if you want a brand new one. By the way, this is the first takedown I've ever had at YouTube, right? So maybe we're getting big enough for people to notice. Yes, total like equals cool. It's the kind of thing that keeps me tuned into microbe TV. You know why some chicken eggs are blue? It's a good story, too. I got lots of good stories because I've been doing this for a long time, and I've accumulated good stories. Here, Manatee has provided a link to Rogan's video with RFK. Click the 3 dots to the right. They'll ask for a timestamp. Thank you, Vanity. This is the kind of help that we need. Now, we got 180 people here. could at least 100 go and say this is medical misinformation, please? Yeah, Lex and Rogan on YouTube. Still there, peeps. Report them. FSM, any thoughts on how bad the UK is with vaccines? I don't know. But we have people from the UK here. Please answer. You can have arguments in philosophy and science without being called misinformation. What dummies censor ideas? Well, there's truth and there's non-truth, right? To say that polio vaccine has killed more people than it's helped is misinformation. To say that vaccines work and prevent disease is true. It's information. It's not misinformation. If you want to say the polio vaccine works by inducing antibodies, that would be an idea. It's an idea that we could discuss, but we don't actually know one way or the other. Sabrina is a fanatical hand washer. I never had COVID. So I hope you use a moisturizer too, because I've seen people who wash their hands a lot and it can really dry out your skin. All right, be careful. Does copper or silver inactivate norovirus? Good question. I think Michael Schmidt has said that copper will. And when copper does, often silver does as well. But I'll ask him tomorrow on TWIM. Okay? Thank you, Tom. Statements about the material medical world that are demonstrably fake are misinformation. That's right. Do you remember what the scientific community's reaction was to the discovery of PCR? Did people immediately realize how useful it would be? Yeah, I remember. People were like, oh my gosh, we can measure tiny, tiny amounts of DNA or RNA and we can use it. Yeah, many people, just like CRISPR-Cas, people said right away, oh my gosh, this is going to be amazing. Yeah. So the scientific community does that. Yeah. Yep. Maybe also a good time to promote TWIV and the Debunk the Funk work on debunking those interviews. Oh, we've talked a lot about on TWIV and beyond the noise about these Rogan Friedman interviews with RFK Jr. So you can check them out there. All right. Nevitz, it's -4 on top of Mount Washington in New Hampshire. Hungry Ghost says President Carter was a naval nuclear engineer. He cleaned up an accident and his urine was radioactive for 6 months afterwards. Not surprising. Yeah. This is good, Spamlet. I'm sure lots of people bite gloves to take them off. Yeah, I don't. But since touch screens and trackpads, it's all fingerless gloves except for purpose-made protective ones. Yeah, I understand. Rob, thank you for your contribution to science communication. Why are there multiple RSV vaccines but no human metapneumovirus? People have been working on RSV vaccines for years. And we'll have metapneumovirus vaccines eventually, yeah. Yes, we're going to have a mini lecture today. Yeah, I'm just getting through a few of these last questions. Pete says all those NVIDIA AI chips are so powerful they will probably outpace quantum computing, which is still lab science. Interesting. Well, fast computers in science are great for solving structures of proteins. You can do it more quickly, more accurately, aligning nucleic acids to find new viruses and so forth. So those are the big computing requirements that'll really benefit. Ray's said, I reported Rogan. 2943 RFK says his kids got 72 doses I posted the CDC link for 32 doses, which I counted on the PDF. Thank you, Rays. Really appreciate it. Starting a click battle with Joe maybe is not a good idea. No, I want to see if that's the mechanism for taking down a video for one person or multiple people to complain. And I want to know if multiple people saying our videos are information or truth, if that helps as well. We're doing science, John. I understand what you're saying. Yeah, Jeff, Lex got into the two sides BS. Yes, he seems to believe various opinions need to be heard even when data refutes an opinion. Like RFA Jr., he has no opinion. He just lies. He just makes stuff up. There's no need to hear him. Of course, yeah, there's no need to hear him. Forget it. The channel Veritasium did a PCR history video about 10 days ago. I found it helpful with the visualizations and the heating and cooling with the polymerase. That's right. That's how it works. Is H5N1 infecting domestic cats? It can, and it can make them very sick. But it's not. It doesn't seem to be spreading from cat to cat so far. So if your cat in the backyard encounters an infected bird, it could be infected, yeah. Okay, let's do a mini lecture. This is facts, folks. This is facts. I should turn off the the last comment there. Okay, back to the screen. All right, we're going to talk about chronic wasting disease today. What is chronic wasting disease? It's a prion disease. And the reason I want to talk about this is because this paper was just published in Emerging Infectious Diseases in January. Detection of prions in wild pigs from areas with reported chronic wasting disease cases, United States. Okay, so these are wild pigs, feral pigs. They detected in tissues from wild pigs living in areas of the U.S. with variable chronic wasting disease epidemiology. The pigs are positive, so they could spread chronic wasting disease. But first, let's talk about what it is. So chronic wasting disease, bovine spongiform encephalopathy, mad cow disease, Creutzfeldt-Jakob, scrapie, Kuru, chronic wasting disease. These are prion diseases. They're caused by proteins that we have within us, no nucleic acids whatsoever. And a huge driver in the discovery and validation of prions was Stanley Prusiner, who got the 1997 Nobel Prize in medicine for that discovery. And somewhere in the world, there's a prion road, which I think is interesting because Stanley made the word up. And here is an article in the Times from 1984. George Balanchine, the director of the New York City Ballet, died of a prion disease. You know, he started having problems walking and moving and deteriorated rapidly and died. That's a prion disease. No cure. These prion diseases are called transmissible spongiform encephalopathies. So an encephalopathy is a disease of the brain. These are fatal neurodegenerative disorders of mammals. Fatal, uniformly fatal. There are thousands of cases a year, and about 1% of those arise by infection, and the rest are genetic and spontaneous. And for example, by 2002, 120 humans had contracted a prion disease called Creutzfeldt-Jakob by eating meat from animals who had a prion disease called bovine spongiform encephalopathy or mad cow disease. You cannot inactivate these prions. It's very difficult. So if you're eating food, you can cook it as much as you want. You'll never inactivate a prion. And there are ways of inactivating prions which are so harsh you can't apply it to food. There are many TSEs of animals. There's BSE, mad cow disease. Then we have chronic wasting disease of deer, elk, and moose. We have exotic ungulate encephalopathy. We have Cats get it, feline spongiform encephalopathy, scrapie in sheep and goats. Mink get it as well. And of course, humans, there are multiple TSEs of humans. There's Creutzfeldt-Jakob, there's fatal familial insomnia, Gerstmann-Straussler syndrome, Kuru, and variant CJ, which is what you got by eating meat contaminated with cow prions from cows who had mad cow disease. Now, what does spongiform mean, right? In the name, transmissible spongiform encephalopathies. The infected brain of these animals or people have a sponge-like appearance. They have holes in it. This is a section of brain. Not supposed to have holes. It's supposed to be solid cells. So the holes are cells dying. You get psychomotor dysfunction, you get cognitive issues, and the symptoms depend on which part of the brain is damaged. Each of the diseases, we had separate human diseases. They each have their own characteristic symptomatology and pathology. Okay, so the first prion disease that we recognized was scrapie. And it's called scrapie because the infected sheep rub on fences. They rub because I guess they're itchy. So the farmers called it scrapie, but they have motor disturbances, uncontrolled trembling, paralysis, weight loss, and they usually die within 4 to 6 weeks. And this has been recognized as a disease of European sheep for over 250 years. It's endemic in some countries. In the UK, 1% of sheep in the UK develop scrapie every year. And you can't eat that meat, right? Because you cannot cook it enough to inactivate the prions. You will get sick. Now, the sheep farmers were key in figuring out what was going on. They found they could transmit scrapie to healthy herds. In other words, they give their buddy down the road an infected sheep inadvertently, and they would infect their herd. So it's an infectious agent. And in 1939, the infectivity from sheep brains was shown to pass through filters that could only pass viruses. But these agents were highly resistant to UV, ionizing radiation, formaldehyde. And the idea arose that they had no nucleic acid in them. They're clearly not typical infectious agents. In 1967, a mathematician, Griffith, suggested that the TSE agents were protein. And in 1981, Prusiner purified infectious protein complexes in extracts from sheep brain with scrapie. He purified the protein, transmitted it to animals, and he eventually called the agent a prion, a proteinaceous and infectious particle. And it's encoded by the PRNP gene, and those genes are essential for the pathogenesis of TSEs, as you will see. So the prion diseases are caused by a protein that we have in ourselves. And sometimes we get versions of it from other sources. So the pathogenic version is a conformational isoform of a normal host protein. So the normal host protein is called PRPC for cellular. It's predominantly on the outer surface of neurons. And the abnormal conformer, which is called PRPSC for scrapie, if you introduce that into the organism, it causes the PRPC, the normal protein, to convert to the pathogenic conformation. All right? So if you eat something with a BSC prion in it, that pathogenic prion can cause the conformation of your proteins, your PRPC, to become PRPSC, and you develop a prion disease. So here's an overview of what's going on. We have PRPC, the normal protein, and here's the structure of that protein, mostly alpha helix, as you can see. If you digest it with proteinase K, it's all chopped up into little bits. This is the assay that people developed to study this. The PRPSC with the abnormal conformation is a lot of beta sheets here, and it is more resistant to proteinase K. You get a 27 to 30 kilodalton fragment when you digest with proteinase K. So this is a way of diagnosing whether an animal has PRPSC in significant amounts. Now, This PRPC can be converted to PRPSC. Well, one way is to just have some PRPSC come in and it converts it. Now, this is the key to the prion hypothesis. And so I'm sorry to give you so much information about prions before we get to chronic wasting disease, but you need to know this. If you knock out both copies of the PRNP gene in mice, you can't give them a prion disease. So you need that gene, the PRNP gene, because if you give mice PRPSC, they have to have something to convert to the pathogenic protein. So I've said that you can acquire a prion disease by various ways: eating meat, getting a transplant of contaminated material. So you'll get PRPSC, and that will cause conversion of PRPC to more PRPSC, and you develop the disease. Some people have mutations in PRNP gene that predispose to spontaneous misfolding of the PRPC to PRPSC, and eventually, whether you acquire it by infection or it happens spontaneously in you, the pathogenic form accumulates in the CNS, and that leads to the symptoms. So 3 types of spongiform encephalopathies. We have infectious, where you're getting some PRPSC by eating or by a medical procedure, by contaminated corneal transplants or contaminated instruments. And that PRPSC causes the conversion of your PRPC to more PRPSC. There are familial or genetic forms where you have a mutation in the PRNP that predisposes it to misfold to form PRPSC. And we have sporadic, where there's no mutations in the gene. You haven't eaten anything contaminated. The protein just spontaneously misfolds. And once you have some PRPSC, it drives the conversion of a lot more PRPC to PRPSC. And all 3 of these can be transmitted to animals because PRPSC is infectious. All right, so human TSEs, there are a number of different ones, right? They're infectious, genetic, or sporadic kinds. We have Kuru. We have people getting prion to TSEs by transplantation of infected corneas, hormones, transfusions from patients with CJD. We have variant CJ, which we got from eating BSE beef. And BSE arose because we fed cows meat from sheep who had scrapie. And the scrapie prion infected the cows. They shouldn't have done that in the UK. Kuru was primarily found in the 4 people of New Guinea with a 30-year incubation period. It was a TSE. And Carlton Gajdusek went and studied it. It found it spread predominantly among women and children through ritual cannibalism of the brains of deceased relatives. So if someone died, the women and children got to eat the brain. And so that spread to Kuru, and Carlton went and he said, folks, we got to stop this. They did, and that was the end of Kuru. Sporadic CJ, 1 to 2 people per million worldwide, mostly 50 to 70 years of age. This accounts for the vast majority of TSCs. It appears with no warning, no epidemiological indications, as what George Balanchine had. They have normal PRNP genes. If you're infected and you don't know it and you say donated blood, you could transmit it to someone else. And it could be that Kuru was established in New Guinea by eating the brain of a person with sporadic Creutzfeldt-Jakob disease. Familial is an inherited disease with mutations in the PRNP gene that predispose them to misfolding, predispose the prion to misfolding. And organs, corneas, blood products for people with sporadic CJ can be infectious and transmit CJD to others. So here are the CJD deaths from 1979 to 2020. We have the number of deaths here on the left, and this is the U.S. This is about 500 here, and the age-adjusted death rate here in red. Now, BSE, bovine spongiform encephalopathy, was basically a form of cannibalism. This is a cow with BSE. It's at the point where it can no longer walk. This resulted from the practice of feeding animal byproducts, including sheep with scrapie, to cattle as protein supplements, so they grow faster. In the 1970s, the method of preparing this supplement, called meat and bone meal, changed and allowed scrapie proteins to survive and pass into cows. And they realized this eventually and stopped, but it took a while for the epidemic to subside. And then people who ate beef from these animals developed a new prion disease called variant CJD from eating BSE beef. So here's the epidemiological curves. Here's the bovine spongiform encephalopathy in orange, beginning in about 88, peaking in the 90s, and then coming down as we recognize we shouldn't be feeding cows parts of scrapie sheep. 1 to 2 million cattle were infected with prions in this time. Now, the incubation period for this disease is 5 years, but cows are slaughtered for meat between 2 and 3 years of age. And so that explains how the prion is able to get into the food supply before any symptoms are evident. Here's variant CJD in people. You see it's overlapping with the BSE. It peaks in around 2000 and then goes down. And then we have a few cases of these individuals who got BSE from eating beef transmitting it to others via blood. So new cases of BSE in cattle still occur. They're sporadic. And we want to protect the food supply, but we don't test all the cattle. Less than 2% are tested. In Japan, every cattle is tested, but not here and not in Canada. We do have diagnostic tests now. We have a kind of amplification test that can be used, and that should be used more often, because it's still possible that some BSE prions get into the food supply. I mean, the cases are rare here. These are the cases in North America. Blue is Canada, orange is U.S. There's a total of 26 between 93 and 2018. So these are cases in cows that have been picked up before the meat enters the food supply. But it doesn't, you only need one to get in, and that causes a big problem. So we're trying to be very careful about this. So I did a podcast with Richard Knight, who's a UK expert on prions, number 950. He knows a lot of things about prions. If you want to learn more, listen to that episode. Yeah, it was recorded in Manchester, UK. So we have had 3 epidemics of Creutzfeldt-Jakob. This is a human TSE. We had one caused by injection of growth hormones purified from human pituitary glands from people who had Creutzfeldt-Jakob. Didn't know it. Transplantation of duromatografts, and variant from eating prion-contaminated meat. Okay. Now, there's one more thing you need to know, and that is there is a prion species barrier. If you inoculate diseased brain into the same species, you get disease, unless you knock out the PRNP gene. If you inoculate into a different species, it's inefficient. So the sequences of the inoculum and the PRPC of the host should be the same. And so the barrier to interspecies transmission is the sequence of the PRP protein. So, for example, if you take hamster prions and inject them into mice, they don't get sick because the sequence is different of the PRP protein. But if you make mice transgenic for hamster PRNP, that is, you give mice the hamster PRNP gene and you infect them with hamster prions, and then they get disease because they have a matching prion protein. Okay, let's take this further. Bovine prions, if you put them into normal mice, they get sick. Bovine prions are odd. They have a broad host range. They can infect many different animals, including humans. We ate cows with BSE, and we got Creutzfeldt-Jakob. So some prions overcome the primary sequence barrier. And that's why BSE is a concern to this day, because it overcomes this barrier. Now, what about chronic wasting disease? This is a recently discovered prion disease of deer, elk, and moose. You can see this deer looks like a scrapey sheep. It's got ruffled, not just ruffled, but abnormal fur, motor problems. And this has been found in 27 states, 4 Canadian provinces, both in free-ranging cervids and in commercial captive cervid facilities. And it's also been found in Norway more recently. So here's a map of the distribution of animals with chronic wasting disease prions. The gray is in free-ranging populations. The dark gray known distribution in free-ranging prior to 2000. And the yellow is in captive facilities, and the red is current captive. So the yellow have been depopulated, and the red are current captive facilities. So you can see pretty far across the U.S. Got some in New York, for sure. And you can see the numbers have been going up, discovered in the 60s, slowly increasing. So we're worried about deer contaminating pastures and other animals picking up the prions, especially if it's a cow or some animal that we are going to eat, right? And that's where this study came in. They looked at wild pigs in states that have been positive for chronic wasting disease, and they have an amplification test that you can use to measure the prion protein. And so they looked at retropharyngeal lymph nodes and submandibular lymph nodes. They looked in the brain. And they looked at a lot of animals. These are feral pigs. The brain, double positive, that is for both forms, 3%, and single positive 11%, negative 85% negative. That's the brain. Then we have the retropharyngeal, 83% negative, submandibular, 62. So obviously, many pigs carry deer prions. This is an amplification assay specific for deer prions. And so the authors of this study say, wild pigs are exposed, although the pigs seem to display some resistance. Future studies should address the susceptibility of this invasive species to the multiple prion strains in the environment. Identification of CWD prions in pig indicated the potential for pigs to move prions across the landscape. So this is the concern here. These feral pigs are mobile. They eat all kinds of things, and they move around, and so they could potentially spread these prions to other places and other animals. And as they spread, they could change and eventually make their way into people. Now, here's what we know so far about the species barrier. If you take cervid prions and inject mice, you don't get disease. But if you make the mice transgenic for cervid prion gene, PRNP, and then inject cervid PRPSC, then they get a prion disease. So again, the prion has to match the recipient, except in the case for cows. Cows can infect almost anything. All right, what about people? If you take mice transgenic for human PRNP and you infect them with cervid prions, you don't get any disease. So this sounds good, right? Well, yeah, as long as the prion doesn't change as it's passing through. It's been found in feral pigs, as I've just told you, and it could pass through other animals as well. So we really need to keep an eye on this to make sure these prions don't get into the human food supply and that they've changed enough to be able to infect humans. So this is a good assay for figuring out whether some isolate of CWD that you have can infect humans or not. So in terms of other animals, if you take deer prions and infect raccoons, meadow voles, and cows, they get sick. Now, this has to be intracerebral, but this prion overcomes the species barrier, obviously. So, deer are pooping out and peeing out prions in pastures, and these animals could graze through and pick them up. None of them have been found to be infected, but hey, we looked at feral pigs, and now we found them. And it's maybe a matter of time before it gets into these other species. So it's definitely a concern that we should be looking at, and people are. Okay. Alrighty. So that I thought you should know about. Chronic wasting disease. All right, let's go to some questions here. Excuse me. Yes, another good reason to be vegan. The students in my class, after hearing this lecture, they say, "Professor, that's it for meat." All right, let's see here where the questions began. Does proteolytic enzyme inactivate the prion? No, it does not. No, it does not. Yeah, Nevitz says prions are an artifact of nature, proteins that happen to fold in a way that lead to pathologies. Actually, there are other prions in other organisms that don't misfold into a pathogenic version, but they do have two altered conformations which have different functions and that are useful. But these mammalian prions have acquired this pathogenic potential. Yes, if you resided in the UK in the years of the BSE outbreak, you can't give blood, at least in the US. I worry about the small butcher shops across the South that butcher wild game. Yeah, so there are precautions that you should take. You should stay away from the lymph nodes, the eyes. You should stay away from the spinal cord and the brain, because that's where most of the prions are. And of course, if an animal is showing signs of a prion disease, you shouldn't take it at all. Vanity, yes. Remember, you said this was your favorite lecture. Yeah. Okay, we got back to the questions. Now let's go forward. I ate half a sheep head, including the brain, decades ago at a New York Italian restaurant. Yes. It's a delicacy. I remember going to a wedding years ago, an Italian wedding, and they brought out a huge tray of sheep skulls with the brains in them, and people rushed up to eat them. But I tell you, it doesn't matter how cooked they are. If they had scrapie, well, I don't think scrapie. I just don't know whether scrapie infects people, because scrapie went into cows, and then from cows it went into humans. So I'd stay away from brains of any kind. Yeah. Good news is that sheep who are genetically resistant to scrapie are easily bred. Yes, but still in the UK, 1% of sheep a year develop scrapie, so that it's not penetrated, right? No, so there's no infectious nucleic acid in prion disease. That's wrong. It is a protein. No nucleic acid is needed. You can produce just the protein with no nucleic acid, and it will infect animals. Yeah. Could someone who butchers and eats wild deer get chronic wasting disease? Well, that's the worry, even though the experiment with transgenic mice that I showed you suggests that humans would not be susceptible. Nevertheless, if you go to there's a website. Yeah, let me show you the slide. If I still have it here. No, unfortunately, I've deleted it. Wait, maybe it's here. Nope. Basically, Chronic Wasting Disease Foundation, or whatever it's called, has a webpage telling hunters how they should dress out deer to be safe. And so far, no human has gotten a prion disease from a deer. Yeah. Are we sure of how prions carry out the pathogenesis? No, we don't know. All we know is that you have this massive protein misfolding in the brain, and this causes this is linked to the pathology, the dropout of neurons and so forth, but we don't exactly know how it happens. However, there are other degenerative neurological diseases like Alzheimer's and others that may also involve protein misfolding and aggregation. So there could be a common theme there. Yeah. Lollipop says, asks, is the inflammatory response with COVID the same as with insulin resistance? I don't know about insulin resistance, but a lot of the brain alterations are probably due to hypoxia and cytokines. Yeah, it's an inflammatory response and leading to long COVID. So identifying which cytokines, right, so your part two of your question, cytokines can damage the brain and contribute to dementia. Yeah, I think that's part of the brain fog and other neurological symptoms that you see in long COVID, because I don't think that I do not personally see evidence that the virus is reproducing in the brain. So how do you explain the neurological damage? Hypoxia, inflammation. Yeah. So the adjusted death rate from prions has not significantly changed over 45 years. That's correct. Pretty steady. Yep. So are the tests for prions based on testing for the prion form? No, they're testing for the misfolded form, right? So what you do is you take a tube and you add normal prions to it. And then you add a little bit of misfolded prion. And then you incubate them. And the misfolded prions will cause aggregation of the normal prions, which you can detect on a gel after treating with proteinase K, right? The misfolded prions have a different digestion pattern. And so if you have a sample that you think has pathogenic prions in it, you add it to a tube with some normal prions and you see if it converts to normal prions. It takes a long time, it's kind of like a protein-based PCR. So it takes a lot of cycles and the tubes have to be shaken and so forth, but it works, yeah. Yes, and as Tom says, they periodically disrupt the growing filaments of abnormal prions, and that provides more pathogenic individual proteins to convert cellular proteins. Artemis, when eaten, how does the prion get from the stomach to the brain? Not clear. Possibly from the intestine. There may be some prion proteins in the intestine, but there has to be some transport to the brain at some point, but no one really knows how that's happening. It's a good question. Vanity says, good grief, scanning the transcript of the Rogan RFK video while listening to you are quite the opposite ends of the spectrum. I'm just a scientist that tells you the truth. That's it. I don't have an agenda. Yeah, I do have an agenda to make you understand science or have you understand science. Tom writes, prions in feral pigs, sinister. Makes me wonder about wild pigs and, say, influenza virus, maybe from migratory waterfowl. Yeah, sure. These wild pigs can be vectors of multiple diseases. And I know that people study feral pigs and susceptibility to influenza, for sure. Yeah. Should do a paper on that in TWIV. Spamlet. It's a shame that knowledge isn't passed on by eating brains. Maybe that was the idea before books. Terrible shame. Lifetime's experience is lost. Yeah. So there are no antibodies or other proteins that can be designed against prion. So actually, good question. Since it's self-protein, we make no immune response against it. We make no antibodies. We make no T cells against prion. So there's no immune response. Yeah. Could you design something? Yeah, I guess you could, but there's nothing. People have tried to make compounds that would inhibit their reproduction, let's say, have failed so far. Has any virus co-infect with prion proteins? That I'm aware of. No, not that I am aware of. It's a good question. Yeah. Yes, Nevitz. And so the mechanism of the disease is aggregation of the prion, not unlike Alzheimer's, etc. Exactly right. Exactly right. If a cervid is positive and grazes on, say, lettuce, could a human who eats the lettuce get infected? Yes. In fact, an experiment was done showing that you can take a plant in a pot in a lab and you put the chronic wasting disease prions in the dirt. They'll be taken up by the roots and go up into the leaves. You can take the leaves and feed them to an animal and they will get a prion disease. So plants can take up and transmit chronic wasting disease prions. Yeah. Yes. Had a surgical patient who needed brain surgery and CJ instruments were destroyed. Yes. You can't sterilize them. There's also a case where a man walked into an emergency room and shot himself in the head, splattering brains everywhere. And then they subsequently tested them and found that he had CJD, and they had to throw out enormous numbers of equipment at high cost because it's very difficult to contaminate. So now there are protocols for this in most ERs and hospitals to deal with prion contamination. There isn't any incidence of scrapie infecting people directly, right? Only through cows. That said, I don't eat sheep heads. Neither do I. Neither do I. Oh, good grief. My brother kills and eats feral pigs. Yeah, you should be careful. Wild boar has hepatitis E also. I interviewed a guy in Germany who liked to hunt wild boar, and they have Hep E. And I said, so what do you do to keep yourself safe? He says, I take the meat and do PCR to make sure there's no Hep E in it. Obviously not available to everyone. Oh, my gosh. Yes, there are other protein misfolding diseases. That's correct. Amyloidosis. Yep. And others. This is interesting. For many of us vegans, we try to stay away from sugar processed with bone char. The U.S. is one of the few, if only, who uses that method, which is ridiculous. Yes. The bones are strangely imported from Pakistan and India. Who's regulating that? It's a good point. Yeah, the DNA, all proteins are encoded in DNA, of course. But the DNA does not have to be transmitted to cause a prion infection, which is a protein that you're eating. All right, so if you eat hamburger, you got DNA in there as well. But it's the protein that is initiating the transformation of normal prion protein. How risky is the cadaver bone marrow used in dental surgery? I don't know. It's a good question. Because various transplanted materials were shown to transmit prions in humans, like, various organ transplants, pituitary hormones purified from human tissue. Even ophthalmologist tools gave people prion diseases because the autoclaving didn't get rid of the prions. So I don't know the origin of those bones used in dental surgery. I'm most concerned about the potential accumulation in soil and plant materials. Yes, that's the study that I just mentioned. Yeah. So bone char is used to whiten the sugar because they thought it would sell better being white. I found out years ago and like, what in the world? Yeah, that's weird, isn't it? Yeah. Is flu vaccine worthless for people over 80? No, I don't believe it is. It has most likely a limited window of maybe 3 months, but usually that's pretty good to get you in the flu season. Yeah. And Cheryl says certified organic is not processed with bone char. Oh my God, the things I'm learning tonight. Yes. Has anyone tried to refold the prion into something less beta sheety? Could that reverse its pathogenicity? Yeah, I mean, I think you'd have to denature and refold the protein, and I don't think that's something that you could do in vivo, right? I'm not aware of any studies that have attempted that. No. Thank you very much, Spartacus. Really appreciate it. Yeah, the gut could deliver it to the brain via the enteric nervous system. Yeah, I actually have a slide that shows that. But it is totally plausible. I agree, but we don't know. We don't know. Yeah, very long incubation period. Totally. Yep. Nevitt says, I predict that with AI plus quantum computing, we'll be able to design a small molecule that will work as an inhibitor of pathogenic prions. Maybe. We certainly have systems to test them in, right? Yep. Will says, off topic, no bats found with close SARS-CoV-2 strain. Could it exist in one of the animals but hasn't been? It could be that it's somewhere in a bat, but hasn't been found, or it's moved on. They have all changed. RNA viruses mutate at high frequencies, so it may have changed enough that it's no longer recognizable. Yeah. In addition, the high rate of pasture being created from forest lands where these infected cervids may have fed, I grow concerned considering the potential for transmission. Yes, many people are. Wouldn't pig Hep E virus be killed by cooking? Yeah, if you cook it enough, right? But also during the butchering of the animal, you can easily get contaminated, right? Because the virus is in blood. So, yeah, that's before you butcher it, he takes a piece of meat and makes sure there's no Hep B in it. Yep, prion origami, exactly right. New York Cohen, what is this from? It says anything that goes against WHO and CDC guidance is misinformation. What is that? What's that from? Yeah, beta sheety. That's cool. I like that term. And alpha helixy. Two good terms. I'm going to use them. Beta sheety. Alpha helixy. Beta sheety. Okay, here's a good question. What does denature of a protein mean? So let's go back to the picture of the prion protein. Now just pick a random one that we can use as an example. Okay. So here's a prion protein. This is the pathogenic one. You see it's got an alpha helix. So the protein is coiled, and then there are these flat portions that are called beta strands. So this is a protein in its conformational state, though it's folded in a specific way. And all proteins have different ways of folding. When you denature it, so you could heat it, and it would become a long string, essentially. You would lose the alpha helices, you would lose these beta sheets, and it would just become a long string, right? And under some conditions, you may be able to lower the temperature, and it might refold into its correct state if you're lucky. It takes hard work to do that. But that's what denaturing means. And you can also denature nucleic acids. If you have a double strand of DNA, you can heat it to 100 degrees Celsius, and the strands will come apart. That's the basis for PCR. Every cycle of PCR, you have to separate the strands of DNA and add new primers so that they can be amplified. I see. YouTube's misinformation policy. Well, we didn't say anything that goes against WHO and CDC guidance, but certainly, yes, it contradicts keeping any RFK Jr. content up because he routinely goes against their guidance. So YouTube needs to be consistent. Oh, you're welcome, Markle. We thank everyone for their donations this season, and I thank everyone for responding to our call for donations. You've been very generous, and you'll all get a thank you from me. I really appreciate that. So, yeah, in my response to YouTube, I'm going to point out that this is not misinformation. Rather, what RFK does is misinformation, and you never take down any of his videos. So it's really an unfair policy. I will tell them that Dr. Offit is a vaccinologist who knows a great deal about vaccines. We publish science on this channel. We don't go for misinformation. It's not what we do. You can ask any of our 134,000 subscribers. All right, folks, looks like you have exhausted your questions for tonight, which is great. Beta sheety sounds like jam sheety, which is the old manual to intraosseous needle used when you can't get a good vein in a patient. Yeah. So doesn't the amyloid protein also misfold via beta sheets and causing aggregation and ultimate Alzheimer's? Yes. In fact, Stanley Prusiner thinks all these degenerative neurological diseases are protein misfolding diseases. Not everyone agrees with him. But, there's another protein called tau that often misfolds and causes tangles leading to dementias. So it could be that that's a common theme. Yeah. Okay, I will look at the language of their policy and quote it in my rebuttal. Thank you. Thank you very much. Yeah, when you cook eggs or meat, you denature the protein. So that's why the egg goes from the non-yolk part. It goes from clear to white because you've denatured the proteins, and when they're denatured, they are white. The yolk and what's the other part? That's not yolk. What is it called? Um, what's the non-yolk part? Egg white. Well, it's funny, or albumin. Egg white, it's not white until you denature it, right? Because it's kind of straw yellow colored in the egg. All right. Anyway, we're going to end this here. Albumin, thank you very much. Now it's coming in. Whites, egg whites, albumin. Thank you very much. I want to thank our moderators tonight for being here. Tom Steinberg was here. We had Les, Fabi. We had Steph SF. We had Vanity Nutrition. Thank you so much for being here. I noticed there were some people that needed to be removed for various reasons, so thank you for doing that. And thanks to all of you for coming. We had 182 people at the moment, 166 likes. We could do a little better in the likes. But yes, hence you can't unscramble an egg. The protein is denatured. Yeah. I don't know, maybe you could put it in urea or something, and I don't know, maybe you could do it experimentally, but you wouldn't want to eat it. But look, folks, please, if you haven't done so already, at the minimum, just go to YouTube's Microbe TV channel and tell them this is not misinformation. This is absolutely factual stuff about vaccines. You're wrong, blah, blah, blah. So thanks all of you for coming in. We'll be back next week. Now we have to stay in touch because we have to see what's going on. I mean, I'm going to record a new Beyond the Noise with Paul on Monday. I don't want to be having to deal with this all the time. So we'll see what happens. We'll give you an update next week. So thanks for coming, everyone. We'll be back on January 15th. That's a week from today. And in the meantime, I wish you all a great week. And be safe until next week. All right? Promise? All right. Goodbye. Goodbye, everyone. Good night.